Hello, everyone. My name is Alexis,
And my name is Morgan.
and our webinar will be about the enzyme Firefly luciferase.
Here is a brief overview of what will be discussed today. We're going to start off by providing a background of what luciferases are, as well as their biological functions. Next, we'll go over the classification and structure of Firefly luciferase. We'll go over our BLAST application and genomic composition and analyzing potential PCR primers. And lastly, we'll analyze the biomedical application of this enzyme.
Luciferases are an enzymatic group that oxidizes luciferin for bioluminescence, also known as light production. This luciferin-luciferase system is found within both aquatic and terrestrial species, like fireflies, bacteria, and jellyfish, just to name a few. More aquatic species use the system than terrestrial species, though firefly luciferases is frequently utilized in the scientific studies.
Biological function of the luciferin-luciferase bioluminescence system has been studied nearly to the same degree as its chemical reaction. Throughout literature, it's agreed that the primary function of luciferase in almost any organism to express it is signaling of some sort, utilizing the light that the enzymatic reaction results in. Some organisms, like brittle stars and jellyfish, use it to deter predators or signal toxicity, similarly to the appearance of bright colors displayed on other organisms. Others distract a predator with a glow or obscure their shadow to divert predator and or attract prey. Luring in prey is another well-known use for bioluminescence. Anglerfish and glowworms are two species to do this. In fireflies, however, luciferase is primarily used to identify and to attract compatible mates of the same species through flashing light. The patterns are species-specific, and when two fireflies synchronize their flashes, it indicates their acceptance of a mate.
Firefly luciferase is found within the firefly species Photinus puralis, otherwise known as the common eastern firefly. This enzyme is classified as an oxidoreductase. Oxidoreductases catalyze the process of transferring electrons from the oxidant to the reactant using oxygen, ATP, magnesium 2+, and a substrate, in this case luciferin. The first reaction forms an enzymatic link luciferyl adenylate, and the second reaction uses the luciferyl adenylate to undergo oxidative decarboxylation. This results in the production of an excited state of oxyluciferin, and luciferase returns to its original state. Oxyluciferin's return to the ground state is what causes the emission of the yellow-green light.
Here's the structure of firefly luciferase. The structure is a 62 kilodalton protein that is folded into two compact domains, the N and C terminal domains, which have a cleft between them. The N terminal contains an alpha-beta alpha-beta alpha 5-layered structure with a beta barrel, while the C terminal contains 5 beta sheets and 3 alpha helices. The domains are connected via a distorted loop, and the C terminal is folded more compactly and is smaller than the N terminal. The literature consistently assumes the cleft closes to surround the reaction, and light emission is possibly due to the two domains closing in to allow the residues and substrates to interact. We chose to highlight how BLAST can be utilized to create PCR primers using imported coding DNA sequences, known as CDS. DNA sequencing of firefly luciferases by the author DeWet et al. The journal article here, Firefly luciferase gene structure and expression in mammalian cells, reveals 6 introns, or non-coding regions, and 7 exons, or coding regions, within the 2,387 nucleotide range. We decided to amplify exon 4, which is located from 1,128 to 1,448 within the sequence, a length of 321 nucleotides. Utilizing the National Institutes of Health's Primer BLAST program, we imported the PCR template M15077.1 created from the previously mentioned sequence of CDS. Without restricting the range of PCR primer creation, the program was run. Upon completion, multiple primer pairs were generated for selection that covered many sections of the CDS. Primer pair 3 was selected in this instance. The forward primer will be discussed first. It is 20 nucleotides long, contains 60% guanine and cytosine, and has a melting temperature of 60.03 degrees Celsius, all within the acceptable and preferred values for a PCR primer. The forward primer sequence, starting at the 5' end, is GTTACCTAAGGGTGT which contains the guanine cytosine clamp recommended on the 3' end. It also contains TAC at the 5' end, which becomes an AUG sequence once transcription occurs, indicating a start codon. The reverse primer is 21 nucleotides long, 47.62% guanine and cytosine, has a melting temperature of 60.00 degrees Celsius, and a sequence starting at the 5' end of TGCGCTTAACTCACCGTTTTG. While not the most ideal, as the 3' end only contains one guanine for the guanine cytosine clamp, this sequence also contains a stop codon, ACT, which becomes UGA. Paired with the earlier start codon, this would be an interesting segment to study to further understand luciferases. The amplified sequence that results from the use of these two primers is 443 nucleotides long, which slightly overextends exon 4 to include the third intron and the tail end of the third exon. The start and stop codons within the selected PCR primer pair are noteworthy, as within the same study included on the previous slide by DeWet et al., it was discovered that partial deletion of the 5' end of the complete sequence resulted in increased luciferase expression, but decreased or eliminated promoter activation. The study of an alternate start sequence occurring later in the CDS may lead to new insight into expression and promoter activation of the genome. If deletion of a start codon later in the sequence causes increased expression and therefore greater bioluminescence without impacting overall transcription, this may be a more reliable way to boost bioluminescence and improve monitoring capabilities in the biomedical field as relevant to luciferase.
Recent research and clinical trials suggest that firefly luciferase can be used in the biomedical field to monitor gene expression. Prostate cancer is one of the most common forms of cancer found within biological men. Studies show that approximately 1 in 8 men will be diagnosed with prostate cancer at some point in their lifetime. Prostate cancer treatment utilizes similar plans to other forms of cancer, but these treatments show limited value for the patient and their well-being. Clinical trials are beginning to find alternate ways for imaging that are more non-invasive when measuring tumor growth. One clinical trial is particularly interested in utilizing firefly luciferase. This research used adenovirus vector known as ADPSA-Luc, which contains the firefly luciferase gene, to measure the gene expression of the human prostate cancer in mice models. For this in vivo experiment, 6 to 8 week old mice were injected with the C4-2 cell line. The C4-2 cell line was created from LNCAP, which is an androgen-responsive human prostate cancer cell line. Both the prostate cancer model and the naive nude mice, which are being used as a control, were injected with either the ADPSA-Luc or the ADCMV-Luc. The ADCMV-Luc is the control for the experiment. Both were injected directly into the tumor site and were given 2 days for distribution. Bioluminescence activity was measured using a CCCD camera every 2 to 5 days. To detect the luciferase expression, an RT-PCR was performed. The ADPSA-Luc gene expression was found within the prostate cells and within the lungs. The data shows higher light emission of the ADPSA within the chest cavity of 3 of the 6 mice at approximately 4.5-fold higher than the naive mice and 8.55-fold higher than the ADCMV-Luc injected mice. The RT-PCR assay detected mRNA of the human prostate cells within the lungs and in the prostate cancer mice and naive mice. The following slides will go in more depth about the results obtained. The image on the left shows the progression of light emission in one mice model that was administered the ADPSA. On day 2, the CCCD camera detected minimal emission within the tumor region. But on day 30, you can see varying levels of light emission both in the tumor region and on the chest area. Day 7 was when the signals within the chest became abundantly apparent. The bioluminescence signal ranged from 6.1 times 10 to the 4 to 3.8 times 10 to the 5 RLU per minute. The image on the right shows the progression of distribution each injection had on the naive and the tumor-bearing mice models. When first looking at the image, you can see that the injection of the firefly luciferase, or ADPSA, resulted in a signalization of tumor regions since hardly any light emission was detected from the naive mice. Results of the RT-PCR will show why the chest area of the mice show light emission. The image on the left represents the CCCD imaging of the isolated organs from the chest, cavity, lungs, and liver. In A, B, and C, you can see the difference of light emission between the lungs and the liver. The tumor-bearing ADPSA group showed significantly higher light emission within the lungs. The results of the RT-PCR, which you can see on the right side of the slide, confirm that the human prostate antigen, known as PSA, was expressed in this tissue. These results weren't expected to come with this experiment, but allowed the researchers to detect lung metastases forming in this region. This study didn't completely provide the results the researchers were hoping for. However, it is a step in the right direction for the medical field in providing a non-invasive and potentially safer option that promotes a better well-being for the patient and their diagnosis.
That concludes our project on the enzyme firefly luciferase. We hope you enjoyed learning about this enzyme.